Okay, so the next topic we're going to be talking about is protein domains. The human genome contains greater than 21,000 genes. The product of majority of these genes are proteins. There are two possible explanations on how these protein coding genes came to be. They were created de novo, as in through random chance a piece of DNA was synthesized that encoded a functional protein. A few genes were created de novo and a majority were made through copy, mutation, reshuffling of parts. Hypothesis 3A would be an unlikely method by which 20,000 protein coding genes came to be. So let us assume we have a unique protein that is 100 amino acids in length, from the N-terminus to the C-terminus. That means, not accounting for introns, the coding region is 300 base pairs in length. There are 4 different DNA bases. Therefore, the odds of a unique sequence that encodes our 100 amino acid protein coming together through chance would be 1/4 to the power of 300. And we can all agree that this is a very small number. So there's a very small chance that if you have a protein that is 100 amino acids in length, that the gene can come together through just random chance. There's plenty of evidence to support hypothesis 3B. For example, the G protein coupled receptor, which is a receptor that we'll be talking about in section 3, are a class of cell surface receptors that are involved in taste, light, neuromodulation, and a lot more. So obviously, this class of proteins, these G protein coupled receptors, they are involved in many different biological responses and systems. All G protein coupled receptors have a very similar structure, a single polypeptide chain which passes through the cell membrane 7 times. So here we have a cartoon drawing of a G protein coupled receptor, and you can see that it's a single polypeptide chain that goes through the cell membrane, this being the cell membrane right here, separating the extracellular space from the cytosol. And all G protein coupled receptors will have this similar shape to it. So we have a ribbon drawing of a G protein coupled receptors. You can see it's very alpha helical. You have many alpha helices, not any beta sheets. And right here is the binding pocket. And this is the molecule that will bind into that binding pocket. So for the picture above, you can imagine that the binding pocket would be somewhere around here. Okay? What is different between G protein coupled receptor is the amino acid composition of the binding pocket and part that sticks out into the cytosol, which interacts with a cellular partner. So let's go back and take a look at this picture. So all G protein coupled receptors have a similar tertiary structure. Their primary structures can be somewhat similar to each other. And of course, their secondary structures are very similar to each other. But the difference is going to be that the amino acid sequence in the binding pocket, which we had pictured, circled right here, right? The amino acid sequence there would be a little bit different between different G protein coupled receptors. Also, this area right here interacts with proteins that are in the cell. So a receptor would be completely useless if the only thing it did, it bound its ligand. Because then the ligand binds to the receptor, and then if that's the only thing that happens, then nothing will happen afterwards. So it's a kind of wasted interaction. So once the receptor binds its ligand, something happens to this area right here in the cell, the cytosolic part. Let me actually put a different color than red. Here we go. And that little bit of a change in the conformation on that side will allow for other proteins that are within the cell to bind to the receptor, and that leads to a cascade of events happening within the cell that eventually leads to some sort of response by the cell. So if we were to look at it, you would say receptor plus ligand will eventually lead to some sort of response by the cell. Okay? So different G protein coupled receptors will bind different ligands. And also, this area right here, this intracellular part, will be slightly different because it will bind to different cytosolic partners to get different responses by that G protein coupled receptor ligand binding. But the blueprint for the G protein coupled receptor is the same. Just change a few amino acids here and there in important parts, and you get a unique G protein coupled receptor that has unique properties, that binds a different ligand, that activates a different response by the cell because it binds to different binding partners in the cell. So how did almost 1,000 G protein coupled receptor genes in humans come to be? And that's a pretty big number, right? 1,000. And they're all very similar to each other. The current and widely accepted hypothesis is a single G protein coupled receptor was duplicated through mutational events. So there are types of mutational events that occur that lead to gene duplication, meaning that, for example, let's say this is my chromosome and I have a gene right here. And I have a number of other genes, but let's just put this gene right here and we'll call this gene 1, okay? And you get a major mutational event that occurs, and what you get is a duplication of that gene. Here we go. Now we have 2 copies of gene 1. You could have 3, you could have 4. Now you might be saying, oh, this shouldn't happen that often, right? It's a common event that happens in cancer, where you have duplications of genes, where you have multiple copies of a single gene. Since these were extra copies, they could be randomly mutated over time without a fitness cost. So if you already have a functional copy, the other copy is not really needed, so it can be mutated. So you can imagine that over time, what will happen is that extra copy, here's the normal copy, that extra copy will start to pick up some random mutations within it. So here's, these red lines are just random mutations. So now instead of being 1, we'll just call it 1' for now. If the duplication event occurred in a certain way where the promoter and all the other regulatory elements of the gene were also duplicated, anytime 1 is made, then 1' will be made too, because they have the same regulatory elements. So anytime the protein product of gene 1 is made, the protein product for 1' will be made. But now, 1', the product is slightly different than 1, right? So you're going to get, through transcription translation, protein 1, and you're going to get also protein 1'. Through these random mutational events, the extra copies pick up new functions that confer a fitness advantage and were selected for. So if this protein 1' right here is a functional protein that confers some sort of advantage to the organism, then this new gene will be selected for. And now you have a new gene in the population, in the gene pool. And you can imagine this is how G protein coupled receptors came to be. You had one G protein coupled receptor coming together through some random events, and then you have that single copy being duplicated over and over again, and those extra copies were mutated, and then you get new G protein coupled receptors with new functionality, and those that conferred an advantage to the organism were kept through natural selection. Another method for making proteins with new functions is through domain shuffling. A protein domain is a polypeptide chain which, if excised from a larger protein which it is a part of, will fold into its tertiary structure and retain its unique function. Very important. Here's an analogy. The protein is a car. Now, the car has many parts that you can take off of it, and that part will still have the properties it had before. Now, it worked better when it was part of the car, but it still has those properties. For example, if I took the wheels off of my car, it'd still be a wheel. It will still function as a wheel. I can transfer that wheel and put it on another car, right? If we look at a gene for a protein, so here's a gene, and you always have a promoter region. So we have our promoter in green, and this is where RNA polymerase binds to. And then you have the coding region. So here's my coding region. Now the coding region, right, could be one domain. For example, let's say this is domain 1. Now we're not putting the introns in, so let's just imagine we live in a world without introns. Then I might have a coding region for a different domain and a coding region for another domain. And here's my gene, starting from 5' to 3'. So my protein that will be made will have, once it's folded in its tertiary structure, let's say that domain 1 right here will look like a circle. And then there's a small linker region. And then you have domain 2, which we'll say is a square. And then you have domain 3, you have a small linker region, and you have domain 3, we'll say it's a triangle. Okay? Now, each one of these domains has some special property or some special function. Now, each one of these different domains is part of a larger protein, and they each bring their special function to that protein. So, for example, domain 1 can have enzymatic activity. Domain 2 could be a binding domain. And maybe domain 3 is a localization domain, meaning it localizes the protein to a certain part of the cell. Now, what I can do is I can remove each one of these domains and they will still do their function. So if I were to excise domain 3 like that, this domain will not lose its biological property. It will still go and localize itself to wherever it was localizing the full protein. Same thing with the enzymatic domain. If I was going to excise out the enzymatic domain, it will not lose its enzymatic ability. It'll still be able to have that enzymatic activity. It's just that now it doesn't have those partners, the binding partner of domain 2, the localization partner in domain 3. So now it's sort of floating around in the cytosol. And although that does not seem important right now, we will see later in the course that localization and binding are extremely important. For example, for an enzyme, the localization domain can bring the enzyme closer to its substrate, and the binding is to actually bind the molecules that are involved in the reaction the enzyme is catalyzing. So you can think of a multi-domain protein as sort of a complicated machine, that you bring these parts together where alone these parts weren't very good, but together now it's doing something very complicated. The hypothesis is that through mutational events, domains are shuffled and new genes are created whose products, the protein they encode, has new functionality. Example, if a gene for a kinase is fused with a gene for a lipid binding domain, a new protein is created which binds to lipids and has kinase activity. A kinase is a type of enzyme, and we've already come across a couple of kinases, hexokinase, phosphofructokinase, PFK. And kinases are always enzymes that do this. They take a protein or a target molecule, they use ATP, and they phosphorylate that target molecule. Now, of course, the kinases are very specific, just like any enzyme, for their substrate. Pyruvate kinase is not an actual kinase because it actually uses ADP to make ATP. I do not know why they call it a kinase, but the name is there. Okay, so sometimes in biology the name does not make sense. So an example of a multi-domain protein is the Src protein. Now the Src protein is an important protein because it's very much involved in cancer. So the Src protein showed above, you have an SH2 and an SH3 domain, and you have a protein kinase domain. So the protein kinase domain is here, and here is the SH3 domain, and here's the SH2 domain. The SH3 and SH2 domain are binding domains, which localize the Src protein to a certain part of the cell and help regulate its function. So many of our proteins in other animals are multi-domain proteins. And here you can see that, okay, we have single domain proteins that are being shown here and here. For example, epidermal growth factor, EGF. And it's telling you what these domains are down here. So let's scroll up a little bit. So we have the epidermal growth factor domain in green. So epidermal growth factor has a single domain of the epidermal growth factor domain. But you can see that this protein, urokinase, and this protein also have epidermal growth factor domains, even though they're nowhere involved in being an epidermal growth factor. Now, the serine protease domain is in brown, so you can see that this protein, this protein, this protein, this protein, they all have the serine protease domain within them. And then there's the calcium binding domain in yellow, and only this protein has it. And the Kringle domain in blue, which this protein has it. Now, the one thing that kind of should be talked about before we move on and end this is that, for example, the SH2 domain is a good example to use here. So let me talk about the SH2 domain. The SH2 domain will bind to a phosphotyrosine. What does that mean? So if a protein has a tyrosine, the amino acid tyrosine, TYR, and this tyrosine has been phosphorylated, an SH2 domain, and here's my SH2 domain, will bind to this phosphotyrosine. Now, every different SH2 domain will not bind to every phosphotyrosine. What does that mean? Different SH2 domains will recognize different phosphotyrosines. That recognition comes from recognizing the flanking sequence in the primary structure. So whatever these amino acids are, so let's say amino acid 1 or prime, amino acid double prime, amino acid triple prime, and amino acid quadruple prime, okay? So one SH2 domain might recognize a phosphorylated tyrosine within the context of one flanking sequence, while another SH2 domain also binds for a phosphotyrosine but recognizes a different flanking sequence. So different SH2 domains will bind to different phosphotyrosines, not all phosphotyrosines. They will recognize a certain flanking sequence around that phosphotyrosine and bind to that. So even though they're the same domain, and the domain, if I'm looking at a protein at the primary structure and I recognize that the primary structure has an SH2 domain, I know that this protein more than likely binds to a phosphotyrosine. But based on that sequence, I don't know what the binding partner is. I would have to do other experiments to find which protein that is phosphorylated at its tyrosine is this protein binding to. So there is slight variability among the domains. Just like even though the tires on my car is still a tire, there are different types of tires, right? Large, big, specialty tires, etc. that have different functions, but it still has that function of being put on my car, on my wheel, to help me drive my car forward. There's a general tendency for proteins in more complex organisms, such as humans, to have additional domains, as is the case with the DNA binding domain protein compared below. So this is sort of a hypothesis. It's in the book. I don't really know how true it is. Here we have, for example, a DNA binding domain, and you can see compared to yeast and worm, the human DNA binding protein in this case has many more domains in it than the other two. Okay guys, we are done with this section, or this part of it. I will talk to you guys later. Bye-bye.